Oh yeah, you ready?
That helps a lot.
Looking good, feeling good?
Cranberry juice that I have read has some good medicinal properties.
Yeah, antioxidant. Like
Clearing the throat and stuff like that.
perfect. Just what we need for right now, yeah.
Yeah.
Well, welcome. Thank you. Thanks for doing this interview with us, yeah. What's your name again? What's your name?
I don't know, I'm almost 94 years old. I can't remember anything.
I know, me neither. Well, we got some questions here from your friend Aziz.
Okay.
And he wanted me to ask you some questions about you, about your life and stuff. Interview you. So, this question is, when you think back on your life's work, what's the first image or moment that comes to your mind?
Truffles.
Truffles, yeah? Okay. Do you remember your first encounter with a mushroom or truffle, and what did it spark in you?
Well, I don't know about the mushrooms because anyone who has the gift of vision would be seeing mushrooms at the first time they were toddlers and could go around looking in the lawn. But truffles is a different thing because they're below ground. And the first one I ever collected was when I was looking for some samples of mycorrhizae Douglas fir and was raking up the little rootless, and out popped a truffle. But I didn't know what it was. Well, I knew it was a fungus, but what fungus was news to me. I got it with help of Dr. Stuntz, my hero at the University of Washington, who pulled out a recent book by Alexander Smith from the University of Michigan about the genus Rhizopogon, and Stuntz correctly said that's what it was. So we keyed it out, and that was my first encounter, personal encounter with truffles, and I've been looking for them ever since.
So you don't remember the first mushroom you ever saw?
Well, it was probably in my mother's cooking.
Cool. All right. Well, what did the forest mean to you as a young scientist? And what does it mean to you now?
The forest?
Yeah, the forest.
My family often went picnicking in Idaho from Spokane, where we lived, with a family that had a bunch of kids. I think they had 8 or 9 kids, and they were all considerably older than I. So we'd go on a picnic and we'd have a big meal and all, and the older kids would all go swimming in the lake and doing older kids stuff. But I was the only little kid. My sister was there, but she was 3 years older than I. And the head of the other family, his name was Gerhard Kempf, K-E-M-P-F-F. And he came to me and he said, "Jimmy, let's go for a walk in the woods." That sounded cool, so he took my hand. I don't remember, I might have been 4 or 5. But we got into this beautiful forest, a nice little trail, and we stopped, and there was a log with a little tree seedling under the big trees overhead. And so he pointed to it and said, "Jimmy, this is a little hemlock tree." And we know it's a little hemlock tree because you see the leaves on a twig are different lengths. And also because it's growing in the shade on rotten wood, because that's where hemlocks like to grow here. And then he told me about other trees as we saw, like the Douglas fir, that were big trees towering above us. I was really impressed with that. And he kept pointing out things to see in the forest. And that captured me. And I got to doing that any time I was around the forest and see what's new. And in the right time of year, I would see some mushrooms. So that led to what I'm doing now.
Who were your biggest influences or mentors in your early career?
I think Dr. Stuntz, Daniel Stuntz. He was the teacher of mycology at the herbarium of the University of Washington. He was an extraordinarily good teacher. He gave wonderfully designed lectures, and he had laboratories that were, all of us could hardly wait to spend the afternoon at the next lab session with Professor Stuntz. And he made the whole business not only seem like a adventure in nature, but to give us very clear understanding of what mushrooms and truffles did in the ecosystem. Why were they important? And that's where I got my first good exposure to mycorrhizae. And then having found my first truffle, that got me keen on mycorrhizae and the truffles. They went together.
So how did you first become interested in truffle research, and what drew you to this mysterious fungal world?
Well, that's a question that just takes what I said for the last question. Build on that, when I changed from being an undergraduate taking classes, I had a master's degree program at the New York State College of Forestry in Syracuse. The reason I went there is because a friend of mine said, "Hey Jim, I got a job in entomology, forest entomology at Syracuse. Why don't you come along?" And since I didn't have any other specific plans, I knew there was a professor there, Professor Sven Olaf. I forget his last name, but from Sweden, where he had done a lot of mycorrhizae research. So I said, okay, and I applied for an assistantship, and I got it, about mushrooms and truffles, to take students on field trips and other fun things, where we learned a lot together, and that just kept me going with more enthusiasm.
Yeah, so what role has curiosity played in shaping your career?
Say that again.
What role has that curiosity played in shaping your career?
Well, I went on for a PhD at the University of Washington, and there my major professor was Stanley Gessel, who was not a mycologist, but he was a forest soils specialist. So he was a good complement to Dr. Stuntz as farmers, not farmers, but farmers. of my future. And then we had a field session, 6 months, at the university's PAC forest, research forest, where we were out there actually looking at fungi in the forest and getting insights on what the fungi were doing. They were more than just interesting things to look at, and sometimes to pick and to eat if you knew how to not eat the poison ones. But what their role was, making the forest, enabling the forest to function in a healthy way.
What was special about your early career moments that shaped your focus on fungi and truffles?
Well, pretty much what I just said, but for my PhD, I had, when at Syracuse, I had learned about an inconspicuous little fungus that didn't form mushroom or truffles. And that got me interested. Well, how is that operating in the forest and what does it do? It's an interesting fungus because it's hyphae and it doesn't form fruiting bodies, but all of its tissues are black, jet black. And amongst the black mycorrhizae that it forms with trees, it forms sclerotia, little round balls of black hard material. And that was how I could get cultures of them. And I developed a way to get the cultures, and that whole xenococcum business is what constitutes my PhD thesis. But I've expanded a lot since then, especially to truffles.
It was xenococcum, right? Xenococcum? Graniformis?
It was graniformis, yeah.
And you collected around Crater Lake, right?
Yeah.
That must have been really fun.
Well, it was, because Crater Lake gave a nice cross-section of habitats from just middle elevation forest to up to timberline. And so I'd go out with my rake. Oh, should we show my rake? We can do that when we have time. But I'd rake up some mycorrhizae, and I had my hand lens, and I'd look at the mycorrhizae, and there were places on the edge of Crater Lake itself were all xenococcum, all black. Usually in a forest, there's a mixture of different mycorrhizal fungi. Some are white, some are yellow, some are different colors. But xenococcum is the only one that is jet black. I just sample the soil and take it back to the lab and fish out the little black balls and use them to start cultures, and then I could do experiments with them in the lab and in the greenhouse.
So, what are you most proud of in your career to date? What are you most proud of in your career?
Well, that's a hard one, because so much of it was interesting and exciting to me. But one of the kinds of exciting experiments that one can't duplicate is to be the finder of a brand new, undescribed mushroom or truffle. Never apparently studied by anyone else in the world. And I had several experiences in that because I had the good fortune to get grants to do some travel. And one of these took me to Papua New Guinea, north of Australia. And there were a lot of amazing fungi in the tropics, both mycorrhizoformers and decomposers of organic matter and so forth. But I was there with some other folks, and we were all looking for New Guinean mushrooms because they had hardly been studied at all. And I came across a big lump in the ground under the litter fall from the trees. There were some eucalyptus there, even though that genus of trees is almost entirely in Australia. But New Guinea is just north of Australia, separated by a strait of water. And so some of the mycorrhizal fungi that go on eucalyptus got into New Guinea. But this one was really special, a big hump in their soil. So I carefully raked off the humus, and to my extreme surprise, there was a big hypogeous, meaning below ground, truffle, and it was characterized by amazing bright colors. It was as big, I would say, as say a softball, which is quite large for most truffles. And its basic color was a bright yellow, streaked with reds and oranges and some purple and some gray. It was such a display of color, far more than I'd ever seen with any fungus for that matter. So I dug it up and I found a few more. Took it back to the lab, of course, and got good pictures, and it was just such an amazing display of color in a very unusually large truffle. I could not find anything like it in the literature. And at the time I wasn't sure what genus it should go in, or even one kingdom. But I kept trying to place it, and I found a place. And the thing is, it's bright color, and when I cut it in half, a young specimen, the interior where the spores are found, started out gray, and then it would be turning darker, sort of gray-brown. Anyhow, the interior also then became more colored as the spores matured. And I had the pleasure of having to describe and publish that as a new species in a new family, or in a new genus. I can't say yet about the family. But I decided to name it the name
Coleocassis polychromis?
Thank you. Coleocassis polychromis. And I still am quite pleased with that name because of its color. The genus name, say it again.
Coleocassis polychromis?
Oh, Coleocassis. The genus name, the Sina, it's a new genus, but this one would, the full name of the species, Coleocassis, was sunshine or sunset. Well, why name a truffle below ground a sunset truffle? But the reason for that emerges in its species name, polychromis, many-colored. So its name means many-colored sunset, in the obvious reference to its brilliant display of colors on the surface and in its center. So that was a discovery that has always given me pleasure. And finding a new species and genus has also been a pleasure, because to see something first that nobody else has ever written about or spoken about, there's no record that anyone had seen it before I did. It's like being a discoverer of a new continent or something in a way. So that was a lot of fun, and I enjoyed doing that. I've had the pleasure of being able to name a fair number of new specimens, new species in general.
When did you learn, when did you realize truffles were more than just a gourmet food?
Well, first off, I learned that a good many mushrooms are poisonous. So you need to be able to identify a wild mushroom with accuracy before throwing it in your frying pan.
You probably learned that later, huh?
No, I knew that from Dr. Stutz.
I mean, you probably realized that truffles were more than just a gourmet food later. You
Yes.
learned more. You learned about how they were ecological powerhouses before they were more of a gourmet food or garnish. Correct.
Right, but I also learned that none of the truffles are understood to be poisonous. They don't all taste good, but they are eaten by people and by animals, as people are animals too, of course. But I mentioned that because all kinds of other animals love to eat truffles. And they find them because the truffles, as they mature, begin to produce an aroma. And at maturity, when the spores are ripe, the aroma gets stronger. And animals, from little mice to squirrels and bears and antelopes and kangaroos in Australia, eat them. Almost all rodents will seek truffles in the forest and eat them. And they're highly nutritious. But what good is that to the truffles? Well, when the animals eat them, they eat the spores, the reproductive cells of a mushroom or a truffle. So the animal eats them, but it digests most of the tissues of the truffle, but not the spores. The spores have an outer hide, outer wall that does not, that resists the digestive enzymes and so forth that happen when something is eaten. So the animals get their nourishment from the whole body of the truffle except the spores, which are excreted and left in little piles of poop, and that's the way the truffles get their spores spread.
Yeah, I think it's chitin, right? Same thing as like exoskeletons of some insects are made of. Chitin.
I'm not sure. It could well be. I don't remember because I've never made a study of that particularly.
But this is a good segue into the next question, because the next question is wants you to share a story behind a groundbreaking discovery about truffles. Well, that was a pretty groundbreaking discovery about truffles, is that a lot of animals eat truffles, and that's how the truffle disperses itself and reproduces, kind of, basically, right?
Well, that was discovered by a truffle scientists like the German mycologist who studied truffles because the King of Prussia wanted to be able to eat truffles at his royal table, truffles that were collected in Prussia at that time. And it was an empire. And they had, prior to that, had to buy truffles from the French. And the King of Prussia couldn't bear that. So he got this professor to...
Albert Bernhard Frank, right?
Thank you. You've done more study on the back than I have recently. Yes, A.B. Hatch. A.B.
Yeah, he was commissioned by the King of Prussia, right? In the, was it late 19th century, right?
Yeah.
Yeah.
I
wanted to figure out how to cultivate truffles, right?
Well, that led to the cultivation of truffles, yeah.
And he went out and he looked at root tips.
He did. He didn't know exactly where to look for truffles, but he had studied what the French do, and so he found likely looking forests with appropriate trees such as filberts and oaks and so forth. And he looked for places that there was some animal digging, like some little creature maybe looking for a truffle. And so he'd rake away the soil there, and almost always he'd find truffles there that the animals had not yet gotten to. Maybe they weren't ripe when the animals first got there. Anyhow, then he made a scientific effort to get new information just by observing carefully what was to be seen in nature. And that's how he came to his understanding that many, many kinds of animals eat truffles, and that where that was going on, there also was deposits of animal feces that contained spores of what the animal had eaten. And there was the evidence right in front of everybody. But he was the only one who really got into it just by watching what was happening, and then by sampling the results of what an animal defecates after it's been feasting on truffles.
That must have been a big challenge for him.
Yeah. Yeah. And he had students that he taught, and went to other countries in Europe and followed up and got similar evidence all over Europe and got more insights from what they were finding. So between him and his students, that's where we could say truffle science really got its start. Before that, truffles were just thought of as a
disease agents,
edible thing that you ate. Nobody knew what it did in the forest.
Yeah, so what kind of challenges did you face in your early truffle research?
Say that again, please.
What kind of challenges did you face in your early truffle research, scientifically or socially, in academia?
That's a big question. Well, I think I have more or less gone through some of what I added to knowledge and science. But as far as edibility goes, it became clear to me that, and from what I read in the culinary literature, that truffles really don't have much of a flavor that we sense with our taste buds. They produce aromas, and these aromas attract animals, and animals really go after them. I talked to a fellow who was doing some studies of flying squirrels in Washington State, and they had the squirrels with a little tiny radio tracer that they could follow with their cameras. And so it's actually printed directly onto a computer. And they saw that the animals the flying squirrels would jump out from a tree. They don't fly, of course, but they glide, and they can glide amazing distances, like, well, I don't know, maybe as much as a half mile if they get in a big enough tree. Anyhow, they had records where a flying squirrel would jump from a tree and glide down to the soil, and sometimes would get down to the soil and stop and start digging. And they'd dig up a truffle, which they had detected by the truffle's aroma, which had diffused through the upper layers of soil and got out into the atmosphere close to where the truffles were fruiting. And the squirrels would go right to that. And the squirrels, flying squirrels, were so good at this, they saw some squirrels who were gliding down stopped gliding at about 4 feet above ground, dropped right to the ground, and started digging, so they could detect the presence of the truffles well before seeing them, and detect them accurately so that some distance away they could go right to where the truffles were fruiting. I thought that was pretty clever of the squirrels, but I expect most animals would have that. So the hunting of truffles for food, they become very popular in Europe and then spread to South America and their popularity in North America and so forth. So people could train dogs to find truffles, get the dogs used to the good aromas, and give the dogs a reward, a food reward at the time. And it didn't take long before the dogs knew exactly how to get a food reward one they particularly liked. That was by tracking the scent. So in that case, the truffle hunter first trained a dog, goes out with the dog, watches the dog. The moment the dog comes to a quick halt and stops digging, that's about the time that the truffle hunter knows the truffle is there, but he has to get it before the dogs get it and eat it. So he'll run over to the digging dog and get the dog excited, but to hold it back from the spot where the digging was. Then the truffle hunter could put his hands in the ground and feel around with the fingers and determine where the truffles were. And those were mostly the edible truffles that, of course, the truffle hunters did. By edible, I mean have a good flavor. I've eaten a truffle or two that I couldn't smell much of an odor. An animal could. An animal beyond me. But the thing in the culinary use of truffles, which can be very expensive, the ones that are considered the premium truffles for eating, can go up to more than $1,000 a pound, which seems crazy, but people who have the money and have a delight in eating truffles make a market. The trouble is, somebody buys a truffle, or even orders one at a restaurant. If they don't know how to cook the truffles, then what they get to eat has almost no flavor at all. Because cooking the truffles, the aromas are gases. They're driven off by heat. And when that happens, the buyer has spent his money in vain, because it's the sense of smell that gives diners their pleasure of cooked truffles. Has to be cooked in oils where the aromas can accumulate. So truffle oil can be found sold in the market, because that's the way they attract animals to eat and people.
Yeah, some of those truffle oil products that you find in the stores, though, those are the value-added products, and that's synthesized stuff, correct? Yeah, I think it's a very delicate process to try to cook with truffles. There have been some old ancient recipes that say to parboil them and whatnot, and things like that, but it's mostly just a garnish, right?
Yeah.
You've conducted research on multiple continents. What did each place teach you about fungi and culture from each of those places? What do you remember from these locations, like Turkey? What was your experience with the Turkish ecosystem and Aziz's project?
Well, in northern Turkey, particularly northeastern Turkey, are mountains with some very interesting forests, and other places too, but that's mostly along that Black Sea coast, is coastal forest. Is where I've collected been to ancient cities and things like that were most interesting.
You work on a project with Aziz in Turkey?
Yeah.
Can you elaborate on that?
By we, I think Castellano was with me, and I don't know, maybe someone else. But we were looking at the hypogeous fungus of the truffles, basically, of different habitats in Turkey, which had not been given a whole lot of attention yet. I think Aziz was about the only one who had done any truffle hunting. Somebody did, a couple others have published some stuff of Turks, I mean.
You want to elaborate a little bit more on your relationship with Aziz too? How you guys met, and a little bit more on your work, the work you've done with him, and other things you might have done with him?
Oh, who was the last one?
Aziz.
Okay. As I recall, our first interaction was when Aziz proposed to come to our lab in Corvallis. And I think he came for a year with his family. That's when they stayed at my house. And his wife wouldn't let me cook. She wanted to do all the cooking, and that was great because she's a magnificent cook. And she could cook turkey specialties and so forth. She also did the house cleaning, so it was a nice thing. And Aziz and I would go out truffle hunting, and well, right around here, really in the Coast Range or in the woods around here, where there's a lot of herbaceous fungi. So that was a good introduction to him on how we go about finding them. And then, since then, a couple of years since then, he had started a truffle orchard. And that was going pretty good. I don't know how it's going now, which is, well, it's the project. Well, then we went into the European part of Turkey. Part of it is in Europe and part of it is in Asia. So we got some collecting done in the European part, some really thick forests here and there. Aziz and his family, after he spent, I think it was about a year here, and then they went back to Turkey, and they invited me to visit them in Turkey, as I recall, for several months of 3 different visits to Turkey. And the last one, Bev came after Aziz and I had completed our mycological stuff, and she and I went to a really nice travel to the ancient cities there. Some of them, like, I'd have to look at a map to remember the cities, the ruins, but places where we could actually walk on a plaza where, according to the Turkish archaeologists and historians, was the same plaza that St. Paul gave some sermons. Do that again with your face.
Continue.
No. I was just enjoying the wiggle of your ears. And other ancient places like there's the city of Morels where a geological formation, I guess volcanic, that produced a whole bunch of columns of basalt. They're shaped and colored even just like morels. And then I guess these were from fumaroles or something from a volcano maybe. And the part in between was volcanic ash and pumice, which over the millennia washed away, leaving these columns, which by freakish events in the landscape there.
You're talking about Cappadocia?
Probably.
I think it's Cappadocia you're talking about, yeah.
Yeah, sounds right.
The triangular kind of tipped pillars. Yeah.
Yeah.
Yeah. You got to go there with him?
Hmm?
You got to go there with him?
No, that's when Bev and I were finishing off our Turkey visit.
Oh, okay. Do you remember anything about your project with Aziz? His project,
Oh yeah.
that you guys worked on? Was that in Turkey as well? Part of it was in Turkey, or was part of it in Turkey?
Oh, Aziz? It was all in Turkey.
It was all in Turkey? Wow.
So far as I know. Now, he took us through some, I think it was pine plantations that had been inoculated. Unfortunately, the supplier of the inoculum provided inoculum of tuber rumali, which is worth very little in the market. And what happened there, it turns out, is that the supplier of the inoculum bought it from a company that was peeling black truffles for pickling, pickled black truffles, as a way to process and have a saleable product from an otherwise almost unsaleable product. So I got to visit those. I went out with Aziz and some of his students that were working there, and an assistant, I think. And it looked like they had a good thing going, except they got this bad inoculum, which was a call to one's was a warning. Don't take an inoculum afterward of the cellar, especially between Brumai, which is worth the next to nothing. Even if you didn't buy it, it's not for anything, for profit making. And yeah, you had to have for the black truffles, the French blacks. Then there was a hiatus between the Turkaloo's visits to the United States when they came back and acquired some property and had applied for and gotten the green card, which is the prelude to getting citizenship.
Yeah, he was facing some difficult times, right, with the president, that current president at the time, right, Erdogan or something, right?
Erdogan. It's spelled Erdogan with a G, but they pronounce the G as a W.
He was like this. He was like disposing professors and intellectuals and stuff, wasn't he, right?
Yeah.
Why was he doing that? Do you know why?
There's a expatriate from Turkey has been living, I think it was in Maine, a Turkish guy, who was there as a refugee because he was strongly opposed to Erdogan and demonstrated strongly to prevent Erdogan to look like he was in with the Turkish workers, but nobody liked him there. 3 repetitions did the trick.
So do you want to tell me a little bit about your time in India and what you did over there?
Well, yeah, I was working with Indian mycologists, some very good mycologists, and we exchanged a lot of information. It was good for both of us. Several of the Indian mycologists came to Corvallis. One in particularly was chief of an Indian lab. I forget in one city, but I went to visit him there. He was a tall, gray-haired, very distinguished-looking man, but very easy to talk with. So he and I exchanged a lot of information about our research. Also, he was a good ambassador from India to the science community because he was very open and always smiling and enjoyable to chat with. So it was good to have him there. And getting back to what I did in India, was to visit a number of forests that were interesting because they were totally different, of course, of any forest I had seen in the United States. And some of them were also used as good sites for growing some of the Indian spices that like to grow in forests. So they were actually covered with forests, but grew in the shade and were a good crop for the Indian economy. I got to visit some other labs that go into some oceanside forests, which had a different type of vegetation. And I and the Indian colleagues raked up a fair number of interesting hypogeous fungi. Most of them new to the Indians because few of them had any experience in looking for truffles. And that's one thing that I was doing to help them, is to show them how we look for truffles in the United States. And the same approach to that in India, when I was showing them, worked just as well there as at home. So that was another good thing. I had time off while there, doing some traveling more as a tourist. So I saw some of the Indian cities that were notable for their palaces and that sort of thing, and special monuments. Talked to a lot of the Indians, and I think that's pretty broad. I mean, it's not a lot of detail, but suffice it to say, I think it was a good thing for both India and for America. So I looked upon it as a good trip to make.
I have another location right here, Africa. You spent some time in Africa doing mycological work?
The only time I was in Africa was when a plane I was on going from Kuwait to London, and it sat down to let off some passengers, but the plane was surrounded with riflemen, I suppose like a National Guard or something, and they wouldn't let any of us who are not destined to go to Africa. We're not allowed to leave the plane, so I have never set foot on the ground in Africa, and I unfortunately can't say much about it.
What about Mexico? Have you been down to Mexico with any of your colleagues?
Well, yeah. A well-known American mycologist got a grant to hunt for hypogeous fungi in Mexico in the company of Mexican mycologists, and we covered quite a bit of country in Mexico. Altogether, I think between the Mexicans and American citizens, all mycologists, professionals and students working together was a very good experience, and we found a lot of hypogeous fungi, most of which had not been reported before from Mexico, and several that were undescribed species. The American who had that grant was Rita Vilgalis, professor at not at Duke, let's see, professor in I guess it must have been Duke University, yeah. And he was a great guy to come along with, well known nationwide, and mycological research, especially taxonomy. He also did a lot of collecting in Australia. I went with him there. And he liked Australia so much that he and his wife were looking to buy a home there to retire to. Anyhow, that's in Mexico we ate a lot of Mexican food, and also we had the privilege of visiting with Professor Gaston Guzman of Mexico, one of the best known of the Mexican mycologists, and he invited several of us to have dinner at his home, which was a great privilege and very entertaining. We talked about all kinds of Mexican things, and we learned a lot, and he learned a lot. And the aftermath of that, or actually before that, I had gotten to know him well. And he and I have jointly published a number of papers on new hypogeous fungi from Mexico. But I had been there actually a few years before this larger contingent of Americans, at his invitation, and I went to Mexico a full summer with my family, and I traveled a lot around Mexico with Gaston to look at hypogeous fungi and different vegetation types. Meanwhile, back in Mexico City, my wife Bev took our kids to a special school for young children, young meaning 5 to 10 years old or something like that. It was a school designed to teach Mexican children to speak and read English. And we got to go to it. It was a private school and rather costly for a child. But they asked us to take our kids there when they learned about us, because the teachers wanted me and my kids to be there so that these children had daily interaction in speaking English and learning it, how Americans would speak English. And that was a very, very good exchange. Guzman took me and a couple of other Mexican mycologists from Mexico City all the way south to the southernmost parts of Mexico, down to the tropical forests. We collected hypogeous fungus on the way and got a lot of good material, and had a great deal of pleasure working together on them. And that gave us a lifelong interest in keeping interactions between Gaston and me. And that continued until just about 2 years ago, where he died in his 80s. But that was a wonderful experience too. And the other thing about these kinds of interactions were carried forth from us often to our hosts. Our hosts often came to our lab first, and we practiced our no secrets policy. We let visitors know everything we were doing and what were the exciting and interesting results of our research, all before it was published by us, but so it could be shared with our visitors. And in only one case did an American visitor violate that trust by publishing something that he learned from one of my graduate students. But aside from that, Gaston and I learned a lot from each other, and it was a wonderful kind of experience. I hope that policy providing some support for international science can continue, because it's for sure in the interests of all the participating countries. Oh, I forgot going to Mexico for the International Conference on Mycophagy, And my good friend Jamie Urey came with me, and he could speak some Spanish, which was helpful to me. Yes, you could. I said some.
The guy who got grant to do research, to do mycological research down in Mexico, was that Dominguez de Toledo? Does that ring a bell?
Say it again.
Was it Toledo that got grant money to do work down in Mexico?
Toledo?
Yeah, Dominguez de Toledo. Or was that Arturo Estrada Torres?
I don't remember.
Do either of those names ring a bell to you?
Not at the moment.
Okay. It says right here, Arturo Estrada Torres and his students collected what seemed to be the slime mold Chanella simplex in Mexico. Says you and him have unpublished data. Arturo Estrada Torres. No, it doesn't ring a bell.
Nope.
All right.
I talked to a lot of Mexican mycologists wherever I could find them.
Cool. Well, I think we can move on to the next set of questions. This section is about scientific philosophy and your legacy. Here's the first question. What do you consider your most important scientific legacy in the field of mycology?
The most significant what?
What do you consider your most important scientific legacy in the field of mycology?
Well, that's a tough one. I think one of the very important ones is the phylogenetic research that involves the identification of genes and chromosomes and that sort of thing that helps clarify relationships between different groups of organisms. It's clarified the train of evolution from mushroom forms into below ground forms that we now call truffles, at least in English-speaking countries. And that enables a lot more understanding of how these truffles came to be, that they are descended from various mushroom groups. And it's the process of evolution that selects for genes that enable a truffle fungus to produce its spores to be spread by being eaten by animals. That in itself was, ecologically speaking and practically speaking, was important in agriculture and forestry, and of course in mycology, especially with reference to the ones that evolved the fruit below ground as mycorrhizal fungi.
How has your approach to research evolved over the decades?
Well, it evolved very early, I guess. Shortly after I was a learned professor, you might say, in the sense that I had already had my education in mycology. And any mycologist is constantly learning new things. even after going to a university, which gets us off to a good start. But I was able to get funding for graduate students, and that was, to me, a very good bit of progress, because in general these younger mycologists to be proved to be very bright and very hard-working young people. They had bachelor degrees and started on master's programs and doctorate programs, and to work with them was great, because the ones I was able to attract to my program and had funding for added a lot to our mycological progress and research. They were all doing very accurate and very important experiments and observations for their thesis. But for their thesis was just almost a side effect, that they got credit to publish their work and go ahead and make it, extend it farther into the science as the time went by. And most of them, I think, with one exception, was a well-advanced students who died from serious disease before getting a position after he got his degree in mycology. So, getting all those young folks into the science, I think, was a good product of our research program. Also because it was characterized by no secrets. Anytime something new and exciting was discovered by a student doing research, would be shared with other mycologists, just like it was with researchers long after they did their college work.
What advice would you give to young mycologists? What advice would you give to young mycologists just starting out?
Do it with enthusiasm, because that's the way people get ahead in science, is to love what they're doing and do it well and work hard at it, but the work becoming a pleasure to them. And that's the way I was taught as a student, and I carried that on to young people who became my students. And except for the one exception that I mentioned, they've all gone on to get good positions and have had a impact on the science in this country.
Yeah, make sure you have a plan, right?
Exactly, yeah.
Make sure you have a plan, make sure you utilize all your resources available.
Do that and look for opportunities. Opportunities for more support, or for jobs that were more to one's hopes. And all that adds to the enthusiasm that's important to for somebody to get involved in science and to want to continue it as long as their life was.
Don't be afraid to ask questions
Absolutely.
or make mistakes.
And not afraid to speak of their own accomplishments so that other people would know what new information they have developed for others to use.
What do you think makes a good scientist or a good mentor?
Well, pretty much what I was just saying. People who have a love of the work they're doing, the science they're pursuing, and looking for opportunities to keep in that profession.
You've had a lot of good mentors through your life, right?
Say that again?
You've had a lot of good mentors in your life, right?
Oh, absolutely. I've mentioned Dr. Stentz already, Dr. Gessel.
Dr. Kessel?
Gessel.
Gessel?
He was my major professor in the PhD.
How do you spell that?
G as in guy. E-S-S-E-L.
And what university was that at? Was that
University of Washington.
University of Washington?
Yeah. A soil scientist, but meaning one who had a special interest in the life of the soil. What is it living in the soil that makes it fertile for plants and so forth? And that, of course, would include mycorrhizal fungi, nitrogen fixing, and a great variety of other things.
A lot of your students are continuing their work out in the field with companies or businesses. How does that make you feel?
What?
How does that make you feel when you see your students continuing their work in the field or with companies and businesses?
It makes me feel happy because all the students I've had, I encourage to work hard, share your information, enjoy it. And when you've made good progress, get it printed, get it in publication. And every scientist likes to see his name as the author of a paper that everybody likes. And again, looking for opportunities.
If you could give a message directly to the mycologists of the future, what would it be? Kind of been answering these questions.
Yeah, well, I've been talking pretty much along those general lines the whole while. But enthusiasm and enjoying what you're doing. An additional good feature, if it works out, is to make a good living at it.
Can you think of a moment when a student or a mentee filled you with pride?
Repeat, please.
Yeah, can you give what's one moment with a student or a mentee that filled you with pride?
With a particular student?
Sure.
Well, let me see. Well, one of the most, one student that gave me the most novel experiences was an Arab student I had from the country of Kuwait. And he arranged for me to go to Kuwait with him because he worked for the Kuwait Institute of Science. Kuwait's a very small but a very rich country because of its oil. And he showed me places where warfare had such bad effects on nature, on soil, on the organisms in the soil that plants need to survive. Without those organisms, including mycorrhizal fungi and so forth, I have a photograph of him standing in what had been a very productive producer of desert truffles, and he'd done studies of them there, studies that complemented other studies that desert truffles grow because they're adapted to the climate, which means that they don't do anything during the drought, but they'll lie dormant in the soil until there becomes a good wetting rain, often from thunderstorms. And that wakes up the truffles to the point that soon after such a staunch rain that came by gallons, that were then made available to other fungi of the same kind. And this made the axiom a good one.
Do you remember that student's name?
Abdul Majid Al-Sheikh. Thank you. Unfortunately, after he got his PhD and he got a good position with the Institute of Research of Kuwait, only then to die of a disease of some kind that affects a lot of people in desert countries, and he happened to get it. And he lived to see much of his thesis published. And he had new studies in process, but he didn't live to finish them.
Aziz's James Trappe National Truffle Institute is inspired by your legacy. Do you have any thoughts on this?
What?
Aziz's James Trappe National Truffle Institute is inspired by your legacy. Do you have any thoughts on this?
Should try what?
Do you have any thoughts on the James Trappe National Truffle Institute?
Thoughts. Well, that's not my choice, but I didn't want to stop it from happening. It was an honor.
Do you have any advice for those building institutions, nonprofits, or research programs in mycology?
Well, be honest. The institutions sometimes are more interested in their own success than they are in the success of new employees. And that's probably not an uncommon thing, but it's very sad to see bright, accomplished young people not getting treated with encouraging policies and so forth that would help both the institution and the students get along and survive and produce new knowledge. That's kind of vague, but I think it happens.
Got about 15 more questions here. These are pertaining to late career and reflections. Some more reflections. What are your hopes for the future of truffle cultivation and research worldwide?
Making institutions do what?
What are your hopes for the future of truffle cultivation and research worldwide?
Well, it's pretty straightforward. You have to learn what is necessary for the truffle fungi to thrive with appropriate hosts. And people like Peter Marshall, a truffle grower in Australia, has had very good success doing his own studies of what these fungi need to be fertile. I was going to need to be happy, but I guess fungi don't go around being happy, but thriving, I should say, thriving. And by careful biological observation and good study design, finding out, the more people finding out about what is required in inoculating trees with desired fungi to get them producing into a way that people can make a living at producing truffles. And the Australian that I know the best about that is Peter Marshall in Australia. And he has designed ways of cultivating and then improving the improvement of the truffle crop. And that's important, but there are some, because it's a big industry now, growing truffles, because they bring a big price in the food market. And there's one book that I'm in the process of reviewing for the Mycological Society of America newsletter. Well, I have,
Taming the Truffle?
Yeah.
By Ian
well,
and...
that too, but also the Truffle Underground.
Oh yeah, by Ryan Jacobs?
And it's hidden. Well, it's a kind of a pun, the Truffle Underground.
Yeah.
But the underground also refers to the criminal industry of stealing truffles or stealing truffle dogs. And
the black market, yeah.
Black market, yeah. So...
and mafia business and gangs and...
Right. Yeah, it's at its highest, so far as I know, as described in this book. It's at its highest in the, I guess, might be thought of as the primary mafia countryside of parts of Italy.
Yeah, maybe you hope that they can start to cultivate some white truffle around here, right?
Sure, yeah. The sometimes called the prince or the king of truffles is the Italian white truffle, tuber magnatum. Magnatum means like the magnificent. And it can bring huge prices in auctions for a variety of reasons. It's often bought at high costs, like some thousands of dollars a pound.
It's like $1,500, upwards of $1,500 a pound.
Yeah.
The last time I read
Yeah.
a book. It's probably
So
probably double that now, at least.
It could be, and that just exemplifies how big money can get in and mess up an industry or even research.
I think that's what it said in Taming the Truffle, but that book is 10 or 15 years. It's been out for at least 10 years now, so yeah.
Yeah.
Prices for white truffles pretty high now.
Yeah.
And that's the Italian white truffle, but we have our own white truffle here. We've got two different white truffles here, right?
Yeah.
Are either of those in cultivation?
Not that I know of. The ones in cultivation in Oregon, so far as I know, are the
Is it Borkii or Lione
no,
or the black one?
the black truffle, yeah.
Cascadiense?
Well, that's a native one.
Carthusiasm?
Well, one of this book up here that I'm supposed to be reviewing for the Journal of the North American Truffling Society. Truffle Hound, yeah. The problem is the Italian white truffle, the most precious one, is hard to cultivate. Nobody's really succeeded in cultivating it in, say, a
anthropogenic habitat.
Appropriate host species like filberts, like that. People who have gotten big success in cultivating the best known European black truffle are getting it to grow in Oregon because it's not as narrow in its requirements of the right environment. But maybe someday somebody will. I think the Truffle Hound speaks of certain Oregon truffles that he has tried and talked to people about to have very high approval in terms of an edible fungus, of its aroma and the good things about it. There may be people trying to cultivate it now. If so, I've not heard of any successes yet beyond the French black.
I think Charles Lefebvre has cultivated the Oregon brown. I think Borkii. I think he's had success.
Well, Borkii is not an Oregon truffle.
Or not Borkii, but Tuberlione, I think, maybe. One of the Oregon brown ones.
Oh.
I think he's had success with cultivating Oregon brown, native Oregon brown ones.
Ah.
Or so he's starting to have some success.
That might be the California.
Yeah, perhaps. I'm not sure exactly right now. How did you meet Charlie?
What?
How did you meet Charlie?
Or Charlie Lefebvre?
Yeah.
Well, I was on his PhD committee at Oregon State University, and he and I have been friends ever since. He's also who invented and carried out the annual Oregon Truffle Festival, and a huge amount of work that he and his wife did to bring truffles into common knowledge in Oregon. Nothing out of me.
Okay, what do you think the world can learn from fungi in the age of climate change and ecological crisis?
I think it's a bunch of bullshit.
No, seriously, what do you think the world can learn from fungi in the age of climate change and ecological crises?
Well, to get a fuller understanding with science in general and with the public on how much the plants and animals of our planet have evolved to depend on what mushrooms and truffles do for them. Particularly, mushrooms and truffles can't produce carbon compounds out of nitrogen fixing sorts of things or carbon fixing sorts of things without getting the energy from the green plant to do it. It's just a sort of a garbled way I just put it, recognizing that what we'd see on any photographs of a forest that has trees can't be there without fungi or grass.
Fungi can't be there without the host trees.
Exactly.
And if you don't have trees, you don't have habitat. You don't have habitat for all these animals.
Exactly.
That perpetuate the spread and dissemination of said fungi, right?
Yeah. But this knowledge is there for the asking. And is it being applied to the crisis or whatever it is the question is referring to? Which in a way, no, I won't say that.
If fungi could speak, what do you think they would say to humans today?
Fuck you. Next question.
This kind of goes back on the last question I just asked you. Not the last question, but the... It says, what can fungi teach us about cooperation, resilience, and hidden networks in nature and society?
To answer that question intelligently, I need maybe 30 minutes to meditate on it.
I think I have a good answer for that question. There's an underground.
Yeah. That's a good answer.
What do you hope the James National Truffle Institute achieves? Where do you see the James National Truffle Institute in 10, 15, 50, or 100 years?
I honestly don't know.
Hopefully providing a lot of research and opportunities for young mycologists, right? Stuff like that.
Oh yeah, well, it hypothetically could do all that, but you gotta have a big bank account to do it.
Yeah, or an orchard.
Yeah.
A producing orchard.
Yeah.
But hey, I think he's got that.
But see, the Nats has a scholarship fund, and it comes from people who pay dues to it.
Mm-hmm.
And the scholarships, I don't know, there's enough every year so far for one scholarship. And I think they're done around $1,000 or something. But sometimes we don't have enough, well, sometimes we have more good applications than we have funding for. So we have to either give less funding to individual ones or give a more useful level of funding to one.
Mm-hmm.
So how the
Prioritizing.
yeah, however, well, whatever. Mycophagy Society of America has a scholarship fund too. But how the Institute will raise funds to do that, I've given no thought to that. And I'm sure Aziz has given thought, but I don't know that he's been able to reach the stage where he can say, yeah, we're ready to go.
Yeah, I see the James Trappe National Truffle Institute around in 10, 50, and 100 years.
Yeah.
Yeah, I see it becoming a pretty big thing up there.
Either that or go bankrupt after 25 years.
Oh, God no. Please no. Hopefully not. Do whatever we can.
Well, you know, Aziz does think big, and he does produce. So.
Ambitious. He's very ambitious.
Yeah, he is that. That's good. Unlike me, who is lazy.
We have ambitious hopes for the James Trappe National Truffle Institute. Big hopes.
Well, I saw an interesting thing on the internet. It's some outfit that has evaluated science and scientists around the world, and in some way, probably by looking at technical papers published or something, what individual scientists have made the biggest identifiable contributions to knowledge, or on down to those that haven't produced anything. And I gotta admit, Jamie, I don't like to talk about this, but it's a fact. I have, so far, published about 510 papers in scientific journals or in books. And I was amongst the people rated. I'm telling you this. I don't want you to repeat it. I don't want to go around broadcasting, hey, I'm a big guy, you know. One thing is that I've continued doing mycological research because it's fun. I love it. And but I've reached the point that most of it I have to do at home, which I can do at home, and I'll keep doing because it's fun.
Yeah, we hope the James Trappe National Truffle Institute contributes lots and loads of good data and publications and research for the next 10, 50, 100, and even 1,000 years.
Why don't I interview you? No, that was very well put.
After decades of studying nature's hidden kingdom, what continues to amaze you?
Booze.
Booze? No, really.
Oh. What continues to fascinate me is to find, well, one thing is to find new species, new genera, new families, whatever, to expand our knowledge of the fungus-sphere. Of course, there are probably thousands of scientists around the world who have a similar interest, which is good. But then I have I get a little bit of questioning about the emphasis that's put on phylogenetic research, that is, looking at the genes and the chromosomes and deciding that this thing is the same as that thing and so forth. But that puts in the background the traditional identification of organisms, fungi in this case, puts it all in the hands to get published, really. Puts it all in the hands of people with the laboratories to do the gene and all that stuff, the phylogenetic. That means the genetics of the groupings of plants or fungi and so forth. So it leaves many of the, I don't know about many, but some of the students very good at the laboratory bench, but not good at having a collection in hands, fresh, getting its physical attributes, its size, its construction, of using a key. And I see in some labs that all the taxonomic work is now depending on the phylogenetic analysis. And I'm not saying that that's bad, that that way of learning about fungi is bad, it's good. But it leaves it in the hands of very few people who maybe never get out in the woods to collect. How do we learn what they're doing out there in that way? I'd like to see, well, we have the Scleroderma, we being particularly me, and Andrew Claridge, and Kaz, whereas Wilson is all laboratory, and that's good because he's doing it good. But then we come up with contradictions. Well, this collection is species A, and that collection that you say is also is species B because it's also the same length of or size of spores or something like that. That can't be published. It's not considered complete. So there are features in our manuscript which we use spore characters more than anything else. Size, shape, ornamentation. And that helps us to say, okay, this species has spores of such a size with spines of this thing. And it's also mycorrhizal with pines. So anything that has those characters, we might put in genus A. And then the phylogenetics people take it, do their extractions, and whatever steps they take in phylogenetics and say, no, this grouping of things because they look alike is no good. They have to have genes alike, but they don't. So the physical characters are not useful without the phylogenetic work. And this troubles me because the work in the lab doesn't really leave us, or doesn't really give to us understandings of, here's this species, we find it by going out and looking for it. And we identify what we find by looking at its characteristics. But how are we to learn about the ecology of that particular one unless it depends essentially on the laboratory analysis, and then find out that the two are not the same in their conclusions? So who wins?
Yeah, it's good to use both morphological characteristics.
Yeah, well,
when identifying in the field, and also using the phylogenetics to resolve such ambiguities. And you're saying don't rely on one or the other, just one or the other.
Yeah. Well, I'm just troubled that so much attention is given to phylogenetic analysis. We're facing this right now, we being the authors of Scleroderma, Hypogeus sclerodermas. Our taxonomy on that basis may all be shot down, or mostly be shot down. So then I wonder, well, what's the point of doing the field work if it's going to get shot down? Or what's putting total dependence on the lab work? Because what they find in the lab doesn't tell us much about what's going on in the field and so forth. So that the mushrooms demystified, or whatever the title is on that, can't be used. Well, it can be used, but it can't be depended on without an equally with equal conclusions from the phylogeny as with the morphology.
Yeah. Yeah, identifying many species by morphology alone, it can change in several ways. Color, size, spore ornamentation. But morphological similarities between some species, they may have been classified under a single name, but phylogenetic DNA has been valuable in resolving such ambiguities, but it's still a complex and intricate process, and trying to rectify field observations with laboratory connections. Still needs some work. Is that what you're saying? Needs a little bit more attention.
Give it equal attention.
Equal attention.
And the problem with the phylogenetic stuff, it doesn't always turn out the same with the same material if you use a slightly different laboratory sequence of study. Well, that was poorly put, but it
basically, lab people, field people communicate.
Yeah. Yeah.
And don't throw out morphological identification methods, and they're useful. They're still useful, and they're important. It's important. It's important to know what you're looking at and what things are, and then but also the genetics are also important too. Like they're important for other reasons.
Yeah.
Anyways,
that
was a tough question. It was.
I guess the way I'd want to put it for an interview like this is that both these approaches are extremely useful and important for identifying things, so we know if we find this mushroom A in Oregon and also in Vermont, and it looks the same on microscopic features as well as macro. But the phylogenetic analysis says no, they're different.
Mm-hmm.
Well, okay, then phylogenists need to tell us in what way are they different? And how does that relate to what they look like?
Yeah.
Microscopically or macro.
Well put.
But I'll never be able to put it again. You missed it.
Oh yeah, we weren't recording. Oh dang it.
Well, by this conversation, I think we can do something about it, not attacking either side, but I face right now that most of the identifications that were made from macro and micro observation, what do they look like fresh? What are the microscopic characters like? Every collection, do they have the same of those? Or does this collection B have shorter spines on the spores or whatever? To be sure that there's oh my God.
Yeah, continue. To be sure what?
It's time for lunch.
To be sure of what? Sorry.
I got distracted by the clock.
Yeah.
What am I going to do?
What will your last words be to the fungi?
Hi, guys.
Bye. You kind of answered this question last night when we weren't rolling, but if you had one final walk in the forest, anywhere in the world, where would it be and what would you look for? You said a mango orchard last night, but I think you were kind of joking.
Yeah, actually, well, it could be in terms of the VA fungi, which might, yeah, mango's probably out of VA, not XO.
Yeah, they are.
So we'd be working just with spores in and around the roots.
But would you want to take a final walk in the Cascades and near Sahican, maybe around Rainbow Falls, or go get back up to Lyman Glacier? Or would you want to be back in Australia? Would you want to be in Turkey or Italy?
Well, what are the goals?
Just to have fun.
Oh.
Africa. You haven't set foot on Africa.
That's right.
Maybe Zimbabwe or.
On me in this event, I will not permit it to be
Utilized?
made public.
Yeah, utilized. That's fine.
You could speed it up a bit.
Yeah.
Well, let me tell you about my interactions with Gaston Guzman in Italy. Well, let me tell you of how Gaston Guzman and I interacted to our mutual improvement.
What's the most unexpected place you ever found a truffle or interesting fungus?
I have looked at, I suppose, tens or thousands interesting truffles. I'm going through them in my mind now so that I can pick the one that was most interesting. At the rate I can do that at 10 per minute. So in 1,000 minutes, I can come to a conclusion. Well, the most interesting one I've already talked about, and that's Siliocassis polychromis in New Guinea. Never seen anything near like that one. And it had me exciting from the very beginning. And it may be a new family even, I don't know. But it certainly is a new genus and new species. I have to give it to the phylogenetic people to see what family it goes in, or what order. But it was startling.
Do you remember a fieldwork moment where something went hilariously wrong or beautifully right?
I'm not so sure about hilarious, but there probably were some occasions. But I would say hilariously wrong. I mean, or just wrong, upsettingly wrong. And that's when we went on a trail to one of Andrew Claridge's special plots in what we all called the Big Study. There were two ways to get there. Up a road that was probably not passable in the vehicle we had, or a trail which was only a couple, 3 miles, as opposed to the road would have been 5 or 10 miles. So we elected to take the short trail, because Andrew knew where it was. But when we got into it, we ran into an area of fallen trees. A big windstorm had gone through there. This was in Mexico, in Australia. A forest had been blown down, and a lot of it was fairly small trees. And since the time it happened, or at least since the time we had last been there, the storm came, and then ferns grew up to about waist high or higher, and just covering the ground with their foliage. And we couldn't see the logs and branches on the ground, so we found them pretty much by hitting them on our shins. And when we got about halfway through, we decided this is enough. We're going to wreck our legs. Let's go back to the road and take it, which we did. We were all limping by then. Our shins were bleeding, but we went ahead and did it like nothing was wrong, just like a Superman would have done. But we got back, we had to bandage our legs, there were scratches and little cuts all over the place. But, well, we were young, it'll heal. Well, it was right, we were young, like less than 25 at the time, or at least some of the students that were with me, less than that. If you count me, you can count that this place, with all the fallen trees, we had to turn around and go back, which we did. By that time, the day was over. It was getting nighttime. And Andrew said, let's get out of here. And we never finished the ideas of why he had chosen that site for, not the windfall, but the site that the windfall trail was taking us. That was important to his research, and we felt badly that we had not done that. But
yeah, you reached Jim Trappe. He can't get to the phone right now. Leave your name and number, and he'll try to get back to you. Thank you.
Well, anyhow, I got to rambling. But anyhow, we ultimately got back to that plot, but a whole day was wasted, and we were all bleeding around the lower legs, which didn't help us over the next few weeks about getting around in the woods, 'cause our legs hurt. But we survived, and we ultimately got back to that plot by the road. And that was that. But I still have some problems with the legs, my legs involved with that hike, because I wasn't sure whether the lights were going to be enough to identify it or to silent.
What about when Efren? Do you remember when Efren fell down a slope in Lyman Glacier and impaled himself on the truffle
on the
fork?
way to Lyman Glacier, yeah.
You want to tell that story?
What?
You want to tell that story?
Yeah, that's a good one. This is part of our study of the role of fungi in the establishment of new forests in the front of a receding glacier where there had been nothing but ice since the Ice Age, I suppose. And so the climate had changed and the glacier was getting smaller, and its front, where it ended, was going back up the mountain that it came from because it was melting. And there was nothing there but ground up sand and soil and boulders and stuff. Nothing to suggest that the important fungi that getting new trees there were missing. And why was it that the older area developed a forest? Because they had none of the necessary symbiotic fungi there. So anyhow, we were going to go up there and check it all out. And the trail to the glacier got fairly steep, along a fairly steep slope. But the trail was pretty much okay, but there was some snowbanks on it. So we were going across the snowbanks, which were sloping downhill at probably about a 30-degree angle. And Efren, who was behind me, just started sliding down the hill. So I ran after him, and he managed to go on his bottom and managed to steer himself around trees. But he landed at a level spot, and he had his truffle fork over his shoulder, and it fell off, and he landed with his bottom onto the truffle fork, and he gave a loud shout indicating that he had endured a accident with the truffle fork. Please help me. So we went down to help him at a faster clip. And there was Efren on his back, but moaning and rubbing his butt. And Efren, we're here. What can we do to help you? Well, he said, actually I sat down when I was doing a bog riot. Graceful fellow. And there we found Efren. He had pulled the truffle fork out of his bottom cheeks.
I thought it was a leg.
Huh?
I thought it was his leg.
Well, the cheeks of his butt. Well, what do you call them?
You told me it was his leg.
This part of the leg is called the cheeks of the leg.
Okay.
But I'll retract that. It had gone into his bottom. And about looked like an inch penetration, maybe a bit more.
That's pretty deep.
And he had tried to pull it out, but it hurt too much because he was reaching behind himself. So I said, well, turn over and I'll try to pull it out straight, which I did. I did it quick so it didn't. So he said, oh, but that was it. And then I put on some tape and antiseptic and so forth. And we decided to go back to camp and go up to the glacier the next day, which we did. And I kept watching the progress of his wound. And it wasn't getting swollen or red or anything. It looked like maybe whatever bacteria was on the truffle fork got washed out by Ephraim's natural defenses. So I don't know where I was going with that. Anyhow, I'm falling asleep.
Basically, Ephraim sustained a pretty severe injury on one of your expeditions, and you guys were far out. You guys are far out. Lyman Glacier is pretty far from anything.
Yes, we were concerned about where we could get for a plane to pick him up if needed.
Yeah.
But as it happened, it just went on and healed, and in a couple of days, he was out walking again.
God bless.
God bless, and he was stoic about it. He didn't complain. In fact, he made a joke of it. So, a couple of days later, we were up on the glacier and looking at it and noticing that while there was a lot of snow still around, because it was a high elevation in early spring, we could see that the end of the glacier was dripping or pouring water from the sun, so it was in the process of retreating. Later on, we noticed that the meltwater and rainwater that fell on the glacier was flowing into its crevasses. It had some big crevasses, and we weren't equipped with ability to rope each other together. We had truffle forks, but we finally concluded that Ephraim was healing and there was not a problem, and that proved to be true. But it made history in the annals of the science of determining how trees got established on the forefront of a retreating glacier. And how we did that, we had photographs of, I tried to take repeat photographs by others who had been there many years before, so we could have before and after on the picture. Well, the ending of that was that we learned from that to be careful when you're carrying a truffle fork over steep snowy ground, and also be prepared, which we were, with adequate first aid material. And 3 summers that we spent up there on very steep terrain often and so forth, we never had another accident. But we also learned from watching the water come out from under the glacier. The water from snow and rain and melting of ice was trickling down the crevasses and coming out the front of the glacier into making a kind of a little lake. And we wondered, was that how spores of mycorrhizal fungi might get into the soil to help trees establish on the newly exposed soil composed of dirt, sand and rocks. So we noticed then a few tiny little.
Do you want to talk a little bit about how you met Chris Mazer and your time and your work with him and collaborating with him on your book, Trees, Truffles, and Beasts?
How did I meet Chris Mazer?
Well, it was you, Chris, and Andrew that wrote that book. I collaborated on that book.
Well, I think I met Chris Mazer through a relative of mine named Doug Urey, because Doug was my nephew and was pursuing a master's degree in use of fungi as food by animals, and Chris Mazer was very knowledgeable, an expert on the small animals around Western Oregon that eat fungi, and one of them was the red-bagged vole, V-O-L-E, cute little mouse. Chris Urey.
You mean Chris Mazer?
Was
or Doug Urey?
Oh, Doug, yeah. Well, through Doug, my nephew, he got acquainted with Chris Mazer, who became his advisor for his master's thesis, and that's how Mazer and I got acquainted and found we had a lot of interests in common, along with Doug. And so Chris didn't know much about mycophagy, and
sorry, can you repeat that last part? I had to pause the video.
What? Well, Chris and I got to working together even after Doug had left with his thesis, which was a good one, and Chris and I started working together about animal mycophagy. Oh, Doug had a captive red-backed vole that he fed a truffle and got a good picture of it. It was live trapping and the hole was in it. Doug had made it look like a natural surrounding and had this picture, which I have a copy of now, a beautiful picture of the vole sort of sitting on its hind legs and with this big truffle in its paws held up to its mouth. This got Chris and me working together on mycophagy and what animals were eating fungi and so forth, and how this was involved in how forests develop, how they begin, and how they develop as they age, and the continuing interactions of fungi, of animals, and the trees that came into focus, that this is a very interesting and very complex system. And this with Andrew Claridge, who was having a Fulbright fellowship at the time, got together to write this book, Trees, Truffles, and Beasts. And that's been, each one of us wrote a particular third of that with interacting between us a while. But it brings together, I think, one of the most understandable picture in words of how forests and truffles and little animals all together create a system that leads to healthy forests. And that's the emphasis of the book. And it's still selling good. So Doug Urey and his, being my nephew, so I knew him well and was keeping in good touch with him and his family because they were good people, good friends, good relatives, and all that is what got Mazer and me and Claridge, who happened to be arriving just about the time I met Chris Mazer. So it was happy conditions. Started with Doug Urey.
I just want to ask you about some other people you know. Randy Molina, how'd you come to meet him?
Say the name is more slowly.
Randy Molina.
Okay. Well, Randy Molina was my graduate student in forestry, and he got his PhD with a minor, I think, in well, I'm not quite sure how it's worked out, but yeah, with emphasis on taxonomy and ecology of forest fungi. And he did a great thesis that was applauded by all who read it about the role of mycorrhizal fungi in the health of forests. And about the time I was ready to retire, he had been appointed project leader for forest mycology in the Pacific Northwest Research Station of the Forest Service. I kept my desk in the Forestry Sciences lab, but he took over all the project leadership of the group, including some that dealt with rare and endangered species. And he retained that until he retired some years later. He kept the same attitude and understanding when he achieved the project leadership and did a wonderful job. I was happy to be there and watch him succeed. He's retired now and no longer is involved in research. He has many other interests to keep him busy in retirement.
I think this is another one of your students, Michael Amaranthus.
I was on the committee of Mike Amaranthus's PhD program, and another faculty member in forestry by the name of Professor David Perry was his major professor. And Dave Perry was a great advisor to Dave, to Amo. And I participated in being a member of his committee as well. Amo was a very enthusiastic student, hardworking, loved working with mycorrhizal fungi in the sense of its importance to forestry. He opened, Emeritus did, his own company, I think called the Mycorrhizal
Applications.
Thanks. And it produced spore inoculum to buyers. He had a very modern, up-to-date laboratory with trained people in it and trained truffle hunters, because spores of truffle species were particularly useful in Mike's view of what was the best for forestry, especially forestry generation. That program and that company continued for many years till Mike was ready to retire, and he decided to sell it, but to sell it only to interested buyers who would maintain the company in its home city, namely Grants Pass, Oregon, and retain most of the workers. And he found a company that was willing to do that, and it continues to succeed in the same fashion that Mike did, and his influence continues in the company.
What can you tell me about Todd Elliott?
Todd Elliott is a student the likes of which I had never encountered, and I'll tell you how I got to know him. I got a letter from him. I had never heard of him. And he said he had found a truffle that he thought he had identified. So I said, well, that's interesting. And he told me the name of it. I forget right now which one it was, but it was one that I knew of. And he was from North Carolina. And I and another fellow mycologist had already named that fungus. So I asked Todd if he would be willing to let me borrow a piece of his find and see if I could agree with it. And I did, he did, and I did, and indeed he had the correct name on it. But in talking with him on the phone, he sounded like, almost like a teenager. So out of curiosity, I asked him how old he was, and I think he was 16. But the amazing thing about this was the letter that he sent me not only had a correct identification, but also had described its habitat and parts of its structure as though he were writing a new scientific paper. So I got his specimen, and it was correct. And he had a complete professional description of it, not only the size and color and so forth, but how the size and ornamentation of its spores, which he could understand only by having a microscope, which he did, and other microscopic characteristics. So I told him that it was a very good paper, and I suggested he offer it to a journal. I think I said mycotaxon, and it was accepted by the reviewers. But it was only then that I really learned that Todd was only 16 years old. But he told me also that his parents lived in North Carolina, and they'd often go to a city in North Carolina that had the Duke University, and they'd drop Todd off, and he'd go up to the office as Professor Emeritus Vilgalis, who was chairman of the mycophagy group at Duke, and well recognized as a top-notch, influential mycologist. And it turns out that when his parents dropped Todd off at the university while they went shopping, they lived on a farm way out in the hills. He introduced himself to Professor Vilgalis and expressed his interest in hypogeous fungi, truffles, that is to say. And Professor Vilgalis offered to offer Todd some laboratory experience if Todd wanted to learn some of that. Todd did, so he kept getting dropped off at Duke while his parents went shopping, and had a good education on how to identify mushrooms and truffles, and what instruments to have, and also the sorts of things that the professional would have at their fingertips. So here was Todd, published a paper at the age of 16 that was accepted by a respected mycological journal, and had been reviewed both by me and Emeritus Vilgalis. Well, this was a startling thing, and Todd and I kept up a correspondence. He kept collecting and finding new stuff near his parents' farm. And over the years, he published more and more papers and got involved with some other professionals, but I was going to go to Australia, so I told him that I would be gone in Australia, looking for hypogeous fungi there. And he inquired if he could accompany me, and I said, well, Dad, yes, you could, but I can't pay your expenses. I don't have any spare money for that. And he said, well, I've been saving money in hopes I could do such a trip. So I said, okay, and we went together. And being young and healthy and very active, he helped me with took care of the luggage and other sorts of things involving the flight and on to Australia. And there he stayed with me for about a month. And I was there to collect truffles. So he did too, and from his experience around his parents' farm, he had good experience. And I'm trying to remember, there was one circumstance where he turned out good at identification of the truffles we were finding in Australia. Not that he knew them well, but he knew how to look into literature about them. Later he came back to America, and he went truffling with me a few times and went up me to the North Cascades and found a lot of truffles in that area in the Sahegan Valley. And by that time he was 18 and had started a university education and went on to become a professional mycology, not only a teacher but as a scientist, at about the age of 25, I would say. By that time he had gone on expeditions with another mycologist to Thailand and also to Africa. So he had cut a wide swath of activity at a very early age and continues now to do long distance travel in search of new and interesting truffles.
Yeah, you guys collected near Ayers Rock in Australia in the desert, right, with the Aboriginals?
Yeah,
didn't you two make a trip out there and study some desert truffles?
Right, thanks. How that emerged was I had met an Australian Aboriginal, as they're called, which Aboriginal is not a put down. It means from the origins of their tribes. And she was working on a PhD at the Australian National University. She was rare as a woman Aboriginal doing that sort of thing. And she and I became good friends, and by that time I had an office near the campus because I was in Australia. I was going to be there for 12 months. And after it came towards the 12-month leaving point, the Australian authorities asked me if I could stay another 4 months, so I said okay. And I got better acquainted with this wonderful young Aboriginal woman who was getting a degree in ecology at the university. But she also took Todd and me, Todd was still around then, to Ayers Rock National Park, where there was an Aboriginal community deeded to them by the government when they changed the ownership of Ayers Rock, this big sandstone ship-like rock emerging out of a flat desert. And they converted the land around it into a national park. The Aboriginal tribes agreed to that because they had the ownership, but the government offered them a continuing ownership of the patch of ground that had good resources of water and so forth, and to help them build a community with a modern hospital and other such things. Anyhow, she took Todd and me out collecting truffles in the nearby deserts, and showed us how the truffle women looked for truffles, and helped us to find our way around. And I've known her a long time since. We've exchanged emails fairly often. So Todd interacted well with the Aboriginal women and took photos of them and me and others out in the desert looking for truffles. Many people hadn't heard of desert truffles, but they do exist not only in Australia, but also in Africa and the Arab nations. Okay.
Todd's a mycological prodigy of biblical proportions, to say the least.
Yeah, I guess it could be put that way, certainly. At his very early age, he was advanced of any young person that I know.
If the mushrooms had to choose an ambassador, I think it would be him.
Yes, I think so.
I think there's another person here I'd like to talk to you, ask you about. David Janos.
David Janos, yeah.
How'd you get to meet David Janos?
Dave is a prominent mycologist. She was a professor at
Miami State? Miami State University?
I'm not sure that was the one. I'll have to ask him again. But anyhow, a university in Florida, and had been on the faculty there, I think, for at least 30 and maybe 40 years when he retired. And when he retired, he left Florida as soon as he could arrange it and moved to Corvallis, Oregon. He had visited here many times to our lab, and he and I became good friends in the process. And when he moved to Corvallis, I invited him to stay in my house because I had space for someone to do so. And he did that and used my place as a home base for searching for a house. And he found something that just met his desires, was able to pay for it, only about 4 blocks from my house. So he and I have continued you might call it neighborliness. I'd go over there once a week usually, and we'd have coffee, and I might bring some donuts or something from the nearby little bakery, and we'd talk about all kinds of stuff, mycology, about other people we knew in common, about strange things the U.S. government was doing, and just anything that came to our minds. And Dave also then had become editor of
the journal Mycorrhiza?
Mycorrhiza. And he did the papers that were about the vesicular muscular mycorrhiza. And so he had a lot of influence on the papers that were accepted for acceptance, even from foreigners whose English was not always very good. But he would take efforts above and beyond most editors would, if he saw real merit in a journal from a scientific standpoint, but badly written, he would rewrite it for the authors, so it would be adequate quality to be published in the journal Mycophagy. So he was one of the most conscientious editors of a scientific journal that I've ever met. And he made it possible for many young and inexperienced or foreign scientists that couldn't write well in English, if their research work was good, he'd make it a great effort. So he'd be working 60 hours a week easily, keep at it. I think he's about ready to retire. Wonder whether he could write someone else's paper again or not.
Yeah, he provides quality editing.
Absolutely.
Quality
Absolutely.
stuff. He did work on Barro, Colorado Island down in Panama too. He studied tropical.
And a lot of work in Costa Rica at one of their big national parks, and he'd take groups of students there and gave them a real field experience amongst snakes and all sorts of jungle things that the kids were not used to, but was a good experience for them.
Yeah, he said that to get to his research station on Barro, Colorado Island, he had to navigate the Panama Canal when there were big ships passing through,
Yeah.
and he nearly got swept away by the waves of a ship that was passing by at one point.
Huh.
So that was a pretty crazy story he was telling me.
Well, the guide of this my interview, Jamie Urey, who, by the way, is the son of
We're gonna cut this out. We're gonna cut this out.
The son of Doug Urey that I had mentioned before.
Yeah. So
And he's more
next question.
he has questions he asks, but he's not as scatterbrained as I am.
You've got some good friends of yours in the North American Truffling Society. Lots of really awesome and great people in the society. Dan and Joyce. You've done some work with them, right?
Oh yeah.
How'd you meet them? Through the society?
Well, Dan was doing a graduate program that was about native fungi to the Northwest. And he got his PhD after a bit, about the time that the North American Truffling Society was started. And Joyce was his wife, but she also was involved in some academic programs. I forget which ones right now. But the two of them have been stalwarts of the North American Truffling Society. And through that common interest with them, we became really good friends, and I've gotten invited to their house for dinner any number of times. And some of those times my interviewer got interviewed too, so he's well involved with the scientific community and especially about truffles.
Well, I think I have one last question, and it's basically over your 60 to 65 years of research in the field of mycology, what have you learned and what has that taught you about yourself?
What have I learned? Everything I can think of about mushrooms and truffles. And I'm not about to explain everything I can think of, except it's been a wonderful experience. I continue it. I retired, I know, about 40 years ago or whatever, just because it's such a pleasure to do that work. It's not work, it's a pleasure. And I'll keep at it as long as I'm capable. So there.
There's always something new to learn, right?
Yeah. And I should also mention my interviewer has also become very interested. Well, I should be allowed to say that. Come on.
What?
Turn it back on.
I didn't turn it off. It's still going.
Yes, you did.
It's still rolling.
Well, anyhow, he's been a real good colleague for me, and he's young and active and strong and energetic, and he's acquired a large arrangement of information in his head about truffles just by continuous reading and self-education, and he's very knowledgeable. And through the experience, which I will say with some satisfaction, I gave him, he's become a top-notch truffle collector. And having become such, I've welcomed his company in going to Australia for 2 different summers where we went for, I don't know, maybe 4 months each year. 2 months each year, it seemed longer. But that's a big story. We had a great time. We were treated well, as always, by friends in Australia. And Jamie has collected a huge number of truffles there.